In this video, we are going to discuss about introduction of recombinant DNA into the host cell. There are several methods are there to introduce recombinant DNA into the host cells. Those are transformation, transfection, conjugation, electroporation through liposomes, direct transfer of DNA, and disarmed pathogen vectors. By these methods, we can transfer the recombinant DNA into the host cells. Transformation. Incorporating of foreign genetic material into the cell through its cell membrane is called transformation. Let us take this DNA sequence. It is a chimeric plasmid. Chimeric plasmid means the plasmid with some foreign DNA is called chimeric plasmid or recombinant DNA, or we can call it as chimeric DNA. Inserting of this chimeric DNA into the host cell through its cell membrane is called transformation. The ability of bacterial cells to uptake DNA from the medium is known as competence. If this bacterium becomes competent or eligible to intake the plasmid from outside, then we can say that this bacterium has the competence to receive the rDNA or chimeric DNA. This transformation can be carried out in ice cold calcium chloride followed by heat shock. What is that? Let us study. This is a normal bacterium. When it is treated with a calcium chloride, this bacterium get a competence to receive the foreign DNA. Here, this calcium chloride neutralizes the negative charges on chimeric DNA and also on the bacterial cells. Because of that one, DNA can attach to the surface of the bacterial cell. Now, the bacterium got competence to receive the foreign DNA into the cell. Now, this bacterium is exposed to high temperature. At 42 degrees Celsius, to this bacterium, heat shock is given. The bacterium which received the heat shock, in that bacterium, the cell membrane becomes porous. The bacterium which undergone heat shock capable to receive the chimeric DNA or rDNA. Due to that heat shock, in its cell membrane, micropores will open. Through those pores, rDNA or recombinant DNA easily passes into the cell. Immediately after putting back this bacteria on ice, the micropores in the cell membrane close. Thus, this host cell receives the recombinant DNA. Such a cell is called transformed cell. In this way, transferring of a foreign DNA into the host cell through its cell membrane is called transformation. The second method is transfection. This is transfection also resembles the transformation. The transformation we use it for prokaryotic cells. If that is carried on plant cells and animal cells or eukaryotic cells, then it is called transfection. Direct intake of naked DNA from the culture medium by eukaryotic cells is called transfection. There, the intake of DNA from culture medium by prokaryotic cells is called transformation. This transfection occurs in plant cells and animal cells. One more method is the conjugation. This is a natural method. It occurs between two bacterial cells. During the conjugation, the bacteria are interconnected by one bridge, one conjugation tube. This is the conjugating tube. And through this tube, the plasmid will pass from one bacterium to another bacterium. The bacterium which donates the plasmid to another bacterium is called donor. The bacterium which receives that plasmid is called recipient. So thus the donor and the recipients they participate in the conjugation. These two are connected by one conjugation tube. After transferring that plasmid, the donor and recipient will separate. Electroporation. In this method, short electrical impulses are given to the cell. This is the cell. To this cell, electric shocks are given. Because of the electric shocks in the membrane, micropores will form. Through these micropores, rDNA enters into the cell. Or vector enters into the cell. This is called electroporation. There is a similarity between heat shock method and electroporation. In the heat shock method, we are exposing the bacterium to the heat shock. Then the pores in the cell membrane are opening. Here, in the place of heat shock, we are giving electric shock. Due to that electric shock, the membrane becomes porous. The recombinant DNA enters into the cell. Gene transfer through liposomes. What are liposomes? Liposomes are the hollow spheres, hollow balls made up of fats. And in these balls, the vectors are enclosed. Or we can say the rDNA, or recombinant DNA, are enclosed in these fat balls are called liposomes. Liposomes fuse with the plasma membrane and the nuclear membrane and deliver the rDNA or foreign DNA into the cell and to the nucleus. In this way, the foreign genes or recombinant DNA can be transferred through liposomes. Direct transfer of DNA into the cells. There are two methods are there here: biolistic method or gene gun and microinjection. In this direct transfer of DNA, vectors are not required in transferring of the gene of interest into the host cell. First, we'll discuss about biolistic method or gene gun method. This is also called microprojectile bombardment. Biolistic method is also called microprojectile bombardment. What is that? It is a shooting of plant cells and fungal cells or animal cells by DNA coated gold or tungsten particles. Is called biolistic method or gene gun method or microprojectile bombardment. In this one, microprojectiles are formed by gold and tungsten particles coated with the foreign DNA. Now, the coated DNA segments are fired into the host cells. In this case, vectors are not used and the gene gun itself is acting like a vector. This method also used in the incorporation of DNA into mitochondria and chloroplasts. And in this way, directly we can incorporate DNA into the host cells and also cell organelles like mitochondria and chloroplasts. This biolistic method mostly used for plant cells. Microinjection. This is also a type of direct method in transferring the foreign DNA sequence into the cell. To hold the cell, they use a pipette, and by observing that cell under the microscope, they inject DNA into the host cell nucleus directly. So the foreign DNA is injected directly into the nucleus using fine needle. This method is employed to inject the DNA directly into the animal cells, eggs, zygotes, and plant protoplasts. The last method is using disarmed pathogen vectors. And let us take agrobacterium tumefaciens. It is called crown gall bacterium and it causes tumors in plants. And this bacterium has a tumor-inducing plasmid. This plasmid has tumor-inducing DNA sequence or TI gene. This gene produces cytokinins. These are a type of growth hormones. These growth hormones cause the tumors when they are secreted excessively. So these plasmids are disarmed by cutting this tDNA, tumor-inducing DNA. In that place, we are keeping the gene of interest and allowing this plasmid or this bacterium to attack the plant cells. Now, this plasmid is transferred to the plant cell and becomes a part of the host cell DNA. The cells into which altered plasmid is introduced are normal, except for the inserted genes. So, plant is normal, but it will express the inserted DNA sequences. So in this way, we can disarm the pathogen vectors and we can use them as mediators to or carriers to transport the gene into the host cell. This method is used for the plant cells.